All right, so I have started recording. So last time we did cell biology. So today we are going to actually do questions relating to cell biology. So that's how we're going to be learning biology. We'll learn the topic, and then next time we're going to do some questions. So you've seen this diagram. It's actually very, very clear. So even the recording will come out very, very clear. So what I want you to do is I want you to write down these questions. I want you to draw the diagrams the way they are. I want you to use the video of today's class to actually answer these questions. So we're going to answer these questions, or I'm going to answer these questions. All you have to do is listen carefully. If you have a question, you can ask me. You pause me and then you ask me. But after this lecture, you go in your biology book, I'll send you the video, you write down these questions, you resolve everything. And then the next topic will be diffusion and osmosis, and then we'll carry on with the questions. Are you ready?
Yes.
All right. So here it says multiple choice. So multiple choice question. So it says choose the correct answer and write its letter in the brackets provided. Question 1 says, study the diagram below and use it to answer the question. So every time you have a diagram, before you even answer the question, I recommend that you first label the diagram. So this is actually a plant cell. How do I know that it's a plant cell? Because it has a regular structure, and you can see that it has two layers. We have got D here, this outer layer known as the cell wall.
Mm.
This structure here, that looks like an oval shape with these three lines. This is the chloroplast. Chloroplast. This structure labeled C is the medium. So this is the cytoplasm. Cytoplasm. And then this cellular structure here, right next to this huge vacuole structure with the nucleolus inside it, is known as the nucleus. The nucleus. So now that we've labeled this diagram, the only thing that we have to do is now answer the questions that follow. So which of the labeled structures contains cellulose? Remember in the previous lecture, I said cellulose cell wall. So our answer is the cell wall, which is structure D. Which of the labeled parts is concerned with the intake of water via osmosis? So I'm going to tell you this here. At the intake of water via osmosis, it's the function of actually the people who it's the cell wall. So the cell wall has something known as root hair cells. Root hair cells. So these root hair cells, their function is to uptake water and mineral salts via the process of osmosis as well as active transport. And by the way, I want to mention this right now that the cell wall is actually fully permeable to water. So the cell wall is fully permeable to water. Water can easily pass through and out of the cell wall. So our answer here is D. In which of the labeled parts is starch made? So the process in which starch is made in plant cells is known as photosynthesis. And last time we said that the equation for photosynthesis is actually carbon dioxide plus water in the presence of sunlight, in the presence of sunlight and chlorophyll to give you sugar or glucose plus oxygen. So that's the word equation. And this process here takes place in specialized structures that have chlorophyll. And these specialized structures are your chloroplasts. Your chloroplasts. And which diagram is labeled chloroplast? It's the C. Chloroplast, it's structure labeled A. So here it's structure A. Are you okay? Are you following?
Yes.
All right, thank you. So let's move on to this question here. Let me get my highlighter so that we read it properly. Two similar amoeba. So an amoeba is actually a single-celled organism with a nucleus, nucleus, and some cell organelles. So an amoeba is a single-celled organism. So two similar amoeba. And these amoeba, they divide via cell division in a process known as binary fission. So this amoeba here, it's going to sort of separate like that, using cell division to make two other amoebas. And the information that's required for cell division is located in the nucleus. So let's go back to the question. Now that we know what an amoeba is, so two similar amoeba were isolated from an experiment. So you isolate it, isolate means capture and observe, probably on a pastry dish of some sort. So two similar amoeba were isolated for an experiment using a fine glass tube. The nucleus of one of the amoeba was carefully removed without causing any damage to the organism. It was observed that the amoeba continued to move and feed for several days. However, during those days, it did not reproduce, so it did not divide via binary fission. On the other hand, the other amoeba reproduced twice for the same period. Which of the following can you conclude about the role of the nucleus in the amoeba? So here we can conclude because let's say this is amoeba 1, isolated, amoeba 2, isolated. In these days, this amoeba 1, they removed the nucleus. So they removed the nucleus for this amoeba. And then this one, they did not remove the nucleus. This one continued to divide. It actually divided twice via binary fission, cell division called binary fission. But this one just continued to get nutrients outside. It continued to get nutrients, but it did not divide. This experiment shows that the nucleus is actually very important for cell division. So based on this, we have to answer this question based on the observation that has been described in the equation. So the nucleus is the only part of the cell that contains DNA. Wrong, because that's not what we are seeing based on our observation. The nucleus controls the normal activity of the cell. No, that's not what we're seeing. Well, we want to answer it in terms of cellular division. The nucleus is essential for cell division, correct. The nucleus is essential for life, no, because we are observing that one bacteria is dividing, the other one is just growing for several days. So our answer here, based on the observable experimental results, the answer is going to be C. The nucleus is essential for cell division. Have you gotten that question or correctly?
Yes.
All right, please make sure you revise this video. Extremely important. Let's move on. So let's look at question 5. This is another plant cell structure, But I want you to observe this plant cell structure. This plant cell structure contains a lot of chloroplasts. If you see a plant cell with a lot of chloroplasts, it means it is specialized. A lot of chloroplast means it's highly specialized for photosynthesis. And photosynthesis is the process in which plant cells make their own food. So it's synonymous with the process of digestion. When we're digesting food, it takes place inside our internal organs. Digestion does not take place on our skin. So the same thing with plant cells. The process of making their own food, this photosynthesis, does not take place on the epidermal layer. So you have an upper epidermis, a lower epidermis, and inside here, known as the mesophyll layer, contains a lot of spongy cells, palisade cells, palisade cells, some air spaces there. So that's the upper epidermis, lower epidermis, lower epidermis. There should be a guard cell there, and this will be your stomata, the airspace to allow air to go inside and outside. So this layer here in between is known as the mesophyll layer, the internal organ of the plant cell. This is where you have a lot of palisade cells and spongy mesophyll cells, and these cells are going to have a lot of chloroplasts so that they can carry out this process of photosynthesis. So this cell here, that's vertically oblong. It's vertically oblong. It means it's like a rectangular structure. It's longer in width than it is in length. So it's shorter in length and longer in width. If you see a cell that looks like this, it's a palisade cell, and it's found in the mesophyll layer that's specialized for the process of photosynthesis. So which of the following correctly identifies the cell and where it can be found in a plant cell? So this is a palisade cell, and it's found in the mesophyll layer that's specialized in the process of photosynthesis. Let's move on to question 6. So question 6, which of the following labeled structures contains chromosomes? So the examiner here wants to trick people. So contains chromosomes. So Fabi, have you ever seen cotton?
Yes.
Ijayo tungi lako yamina ija cotton. It's sort of like, let's say cotton, if you finish it, it's like a woodenish thing. And then there's like threads. Let's say this is a black cotton. So that's the thread like so. That's the thread. That's like a thread like so. So let's say this is the nucleus. That's the nucleus. The nucleus contains DNA. So DNA is like a thread. So DNA is like a thread. It's actually a very, very long thread. It's very long. So for it to be compact, for it to be fitted into the nucleus, it forms a cotton structure of some sort. So there are proteins known as histone proteins, proteins that sort of round around the DNA. So the DNA will start rounding around these to my histone proteins. It will keep on rounding, like so. Then it gets tied there and it round to make a structure known as a chromosome. A chromosome. And this structure here can be packed inside the nucleus. So the nucleus is going to get this same chromosome and it's going to pack it inside the nucleus. So the nucleus contains condensed DNA, wound around these chromosome structures. So every time you see DNA, just think of a cotton, and the DNA is the thread. And if you round it like that, you're going to make a chromosome structure. And this chromosome structure can be contained in the nucleus. And which diagram is the nucleus here? It's structure D, because you can recognize the nucleolus there and the nuclear envelope there. So the nucleus, structure D. So our answer here is D. Are we together?
Yes.
All right. Make sure you watch this video. I'm not dishing out this information just for you to go sit down and not study. You better watch this video, study, understand, pass biology. Let's move on to question 7. Um, which of the following structures are found both in animal cells and in plant cells? I told you this question is going to come. So they are saying found in both. Remember, I told you both plant cells and animal cells need something known as pro
Seven.
protoplasm. Yes, exactly. Remember protoplasm, the living part of the cell. You need a nucleus, you need a cell membrane, and you need a cytoplasm. So these structures here, a nucleus to control all cell activities, a cell membrane that is selectively permeable, and a medium where your organelles can be and all your cellular reactions are supposed to be, and the cytoplasm. So whether you are a plant cell, whether you are an animal cell, you're going to have a nucleus, a cell membrane, and cytoplasm. So cellulose, cell wall, no. False. Chloroplast, no. Vacuole, last time I told you plant cells and animal cells have vacuoles, so this is false. Our answer will be cell membrane. You have understood that?
Yes.
All right. So where are we? I haven't taught you microscopy. What is the major advantage of using What is the major advantage of using a light microscope instead of an electron microscope? So the biggest advantage here is that if we're using a light microscope, the energy source that a light microscope uses is light, but the energy source that an electron microscope uses are electrons. Electrons.
Okay.
So before you view a cell on an electron microscope, it has to be killed and stained, sort of coated with conducting materials that are going to attract the electrons so that you can get a detailed, more structure of that cell. However, for a light microscope, you can just get your slides, add your specimen, get a dropper, maybe add like 1 drop of water to have some medium, and you can view alive cellular structures. You can view the cell without killing it. That's the major advantage. So it's able to observe living matter. That's the biggest advantage that a light microscope has over an electron microscope. You can view the cells while they are still alive. So it's able to observe living matter. We're okay?
Yes.
All right. Okay, I'm glad we're okay. Let's move on to question number 9. Just rubbing the board, clear some space for me to talk and talk, explain stuff. Isn't biology nice?
Yes.
It's interesting. Huh? Yeah, exactly. So question 9, which of the following is a characteristic of mitochondria? Which of the following is so remember, mitochondria, we said They carry out cellular respiration. Cellular
Yeah.
respiration. And the equation for cellular respiration is sugar plus oxygen is equal to carbon dioxide, CO2, plus water, plus energy in the form of ATP, adenosine triphosphate. And because cellular respiration uses oxygen, it becomes aerobic respiration. Aerobic means it's utilizing oxygen. Aerobic respiration, oxygen dependent type of respiration. So that's what the mitochondria does. This is what the mitochondria does. It's the powerhouse, releases energy. What's the equation? Sugar plus oxygen is equal to carbon dioxide plus water plus energy in the form of ATP. Energy, oxygen, it's an aerobic process. So let's go through the multiple choice question. They provide sites for anaerobic respiration. Anaerobic respiration. This is wrong. So every time in biology, if we put an aerobic, it means respiration without oxygen. An aerobic, the respiration is aerobic. Inside the mitochondria, we are using oxygen. It's not an aerobic. An aerobic means the absence of oxygen. So this becomes wrong. They produce cellular secretions. They do not provide any type of cellular secretions. They are responsible for cellular respiration. They store enzymes. Not necessarily the best answer. They do store certain enzymes for a process that I won't mention because you're in high school. It's called apoptosis, but I'm not going to explain anything. There are some enzymes that are carried out in the mitochondria for apoptosis. I won't say that, but your answer has to be they release energy, ATP, energy, cellular respiration in the form of ATP. So our answer will be this one there. Well, okay.
Okay.
So question 10. Which of the following contains the highest concentration of RNA? Remember, I told you the nucleolus is specialized for producing RNA, which is ribonucleic acid. So you have to go through that video on cell biology to understand the function of the nucleolus. It produces ribosomal RNA. So your ribosomal RNA. So it's this RNA, ribonucleic acid, that will be used in the process of protein synthesis, making proteins inside the ribosomes. The ribosomes. So this RNA, it's called ribosomal RNA. It's used in the process of making proteins known as protein synthesis that takes place inside the ribosome. So the ribosomes are a site for photosynthesis. So this marks the end of our multiple choice questions. Let's look at some of the theory questions that we have. This connected to this question here. All right, so let's label this diagram, then look at some of the questions. So just memorize how this diagram looks like. It looks like an animal cell. It's actually an animal cell because there is no cell wall there, so it's an animal cell. So this structure here, sausage-like structure, this is your mitochondria. Your mitochondria. This structure here is they look like stacks. So this is your Goji apparatus. And this structure here is your cytoplasm. And this part here is your cell membrane. So we've labeled all the cellular structures on this diagram. We've gotten marks because the question here says name the structures labeled A to D. So we've labeled all the structures. I think so A, B, C, D. So let's just go back. Where am I? So we said A, Goji apparatus, B, mitochondria. So Goji apparatus, B, mitochondria. Then C, we said what was C? C, cell membrane, D, cytoplasm. C, cell membrane, and D, cytoplasm. Then identify the part of the cell that is responsible for each of the following structures. So stores the genetic information. The structure that stores genetic information is the nucleus. Generates energy, ATP. No wonder I told you the examiner won't say energy. The examiner will say ATP, adenosine triphosphate. No wonder I kept on telling you cellular respiration produces energy in the form of ATP, which is adenosine triphosphate. So generation of ATP from the breakdown of glucose, cellular respiration, aerobic respiration that I just explained. This happens in the mitochondria. Then production of proteins from amino acids. Production of proteins from amino acids is protein synthesis. Protein synthesis. So protein, making proteins, which are amino acids. Synthesis means to make. And this process here, the process of protein synthesis, takes place in the ribosomes. It takes place inside the ribosomes. So the answer to this one is the ribosomes. Where's my annotator? Ribosomes. Well, okay?
Yes.
So let's look at question 2. Question 2 says the diagram below shows a cheek cell from your cheeks, from your mouth. So the question says, so it's an animal cell. The question says, identify the parts labeled X and Y. This is easy. This is definitely X is the cell membrane, right?
Yes. And why nucleus?
Amazing. Wapasa kudala biology. Nucleus, we're doing good. And then draw and label 3 extra structures that would change this cell into a plant cell. So 3 extra structures that would change this cell into a plant cell. So what are we going to draw? The first thing that we're going to draw is a cell wall. So we're going to draw a cell wall. The next thing that we're going to do is draw a large centrally located vacuole, very large vacuole. Then the third thing that we're going to draw is a chloroplast. and we're going to indicate it with 3 lines. So I'm going to say chloroplast, chloroplast, a cellulose cell wall, cellulose cell wall. You can say cell wall, but this part is just to make the person marking you feel nice. Then here you can say a large vacuole. So these things, large vacuole, cellulose cell wall, it's to make the examiner feel nice, because they know that in a plant cell, the vacuole is very large and centrally located. So large vacuole, and they also know that the cell wall contains cellulose. So make the examiner feel happy. Cellulose cell wall, chloroplast, and a large centrally located vacuole. So name the process in which green plants produce stored energy. So name the process in which green plants produce stored energy or food. It's photosynthesis. Photosynthesis is the process in which green plants produce stored energy. So photosynthesis. Where does energy for this process come from? Where does the energy for this process in A come from? So it comes from the chloroplasts. Chloroplasts. So the chloroplasts, what they do is that they convert this sunlight energy they use the sunlight to generate energy. So actually, where does the energy for this process in? It comes from the sun. Then the process takes place in the chloroplast. So sunlight. Let me just write the equation for you. It says carbon dioxide, CO2, plus water
Plus.
in the presence of sunlight. In the presence of sunlight, why do I keep on spelling sunlight with a G? Sunlight and chlorophyll to give us glucose plus oxygen. So the energy comes from sunlight energy. So in what form is this energy stored? The energy is stored as glucose. So the energy is stored as glucose. So you can say glucose. But excess glucose, we even tell them excess glucose is stored as starch. So this energy is stored as glucose, but Vikapaka, when there is too much glucose, they will convert that glucose into starch, which is a polysaccharide. You're going to know what a polysaccharide is, what a monosaccharide is, what a disaccharide is when we do a topic called biomolecules. What are the characteristics of carbohydrates? What are the characteristics of proteins? What are the characteristics of amino acids? You're going to see all these structures properly. So that's how you answer this question. All right, so let's look at some form of essay types of questions. Is this the last question? Yeah, it's the last question on the last question, which it seems long. All right, not so long. So write your answers in the spaces provided. So it says, explain how the limitation of a light microscope in revealing cellular details is overcome by the electron microscope. So explain how the limitations of a light microscope in revealing cellular details is overcome by the electron microscope. So in a light microscope, we're talking about the resolution. So the resolution of, you have to write down this answer uh, when you get the notes. So the resolution of a light microscope, a light microscope is low. Therefore, an electron microscope, using electrons, to scan the surface. Think of an electron microscope as a photocopy machine. It sort of passes electrons on the surface of the cell to produce a very detailed structure. But the bad part is that that cell has to be dead for you to see the detailed structures properly. So therefore, an electron microscope uses electrons to scan the surface of the cell to produce a more detailed, high resolution image of cellular structures. So for example, in a light microscope, you can observe mitochondria like this. And you're like, okay, so this is mitochondria. But on an electron microscope, you can actually see the double layer of the mitochondria, and you can also see something known as cisterns of the mitochondria. You can see all these cellular structures very clearly using an electron microscope. So where are we? So we'll cancel this. Then the next question says, describe the structure and function of the endoplasmic reticulum. So describe the structure and function of the endoplasmic reticulum. So here, remember, there are two types of endoplasmic reticulum. There is a smooth endoplasmic reticulum, and a rough endoplasmic reticulum. Smooth endoplasmic reticulum and rough endoplasmic reticulum. So remember, the rough endoplasmic reticulum contains ribosomes, and the smooth endoplasmic reticulum does not contain ribosomes. So no ribosomes. So in terms of describing the structure, whether it's a smooth endoplasmic reticulum or a rough endoplasmic reticulum, let me write it in essay form now. Whether it's a smooth endoplasmic reticulum or a rough endoplasmic reticulum, you say it's a system or a network of connected membranes. So we're describing a structure that looks like this. So you can see that this is like a system of connected membranes. So describe the structure: endoplasmic reticulum. Endoplasmic reticulum is a system of connected is a system of connected membranes with gained marks by describing the structure. Then we're going to further on and say the smooth, you can just say ER, the smooth endoplasmic reticulum. The examiner will know what you're saying as long as you open brackets and put ER there. So the smooth ER has no ribosomes, its function is detoxification and producing lipids. It also produces lipids that are needed in the cell, so lipid production. Then remember, the rough endoplasmic reticulum contains ribosomes, and I said that in the ribosomes, proteins are made, but they are immature. So the rough endoplasmic reticulum is going to modify the proteins. So the rough ER modifies proteins produced by the ribosomes. All right. And here it says, explain the role of the Goji apparatus. So here we've got the nucleus. I'm going to explain what I explained last time, the nucleus. Then the nucleus is surrounded by the rough endoplasmic reticulum with ribosomes. Then just outside the rough endoplasmic reticulum, you have got stacks. They can be 3 in pairs, sometimes they are 4, they are stacks like that. So the nucleus is going to tell the cell, make stuff, make proteins, and then the ribosomes will make those proteins. So the ribosomes will make proteins. After the proteins are made, the rough ER is going to modify those proteins. And then the Goji apparatus is going to package those proteins, get those modified proteins, so the protein is inside there. Get the protein, package it into transport vesicles, and deliver the proteins in the inside of the cell or the outside of the cell, inside or outside of the cell, where they are needed, They are needed. So the function of the Goji apparatus is to package proteins in transport vesicles and deliver them to various sites. So this will be enough to gain marks because it's grade 12 level. We're not going to say things like endocytosis or exocytosis. We're not going to say that. You just have to say the Goji apparatus packages the proteins in transport vesicles and they will deliver them to various sites. So remember this flow. Always remember this flow here. The flow starts from the nucleus. The nucleus is like, all right, make proteins. Where are you going to make those proteins? In the ribosomes. Where are you going to modify those proteins to make them ready for their functions? The rough endoplasmic reticulum. Where are you going to package those vesicles? The Goji apparatus. That's the flow. So this was a detailed revision of cell biology. So I'm going to send you this video and I want you to write down all the questions with all the answers that we've discussed today. No resting. This is your life that we're talking about. You have to study, you have to work very hard. Then we're going to meet next time. I think next week, the day that we're going to do mathematics, we're going to do an intense mathematics study session. So this is the end of our lecture. I'll send you the notes. Make sure you write the notes, listen to the video, watch it over and over again. I will see you in the next class. Is that all right?
Okay.
All right. See you in the next class. Let me just send you the notes. All right. Bye.
Okay.